Hi everyone, this is a lesson on polyallelism, which is also known as multiple alleles. So let's get started. This one is an inheritance pattern that's kind of complex compared to what you've done before. So polyallelism, or multiple alleles, is where a gene is controlled by greater than two alleles. So up to this point, all you've dealt with is two alleles influencing a trait. Okay, you either had the dominant or the recessive, or you had a pair of codominant alleles, or a pair of incomplete dominant alleles. This time, what makes it more complicated is that you've got three alleles that we're going to look at here for controlling a trait. Okay, and the classic example, the one you're always going to see, is human blood type. So this is how it's going to show up on the EOC. Human blood type is influenced by the gene I, and I know you weren't expecting me to say that, right? Because you know that the types for blood are A, B, and O. Now, some of you right now are saying, well, what about the plus minus thing? What about the A positive, O negative, whatever? That plus and minus is another gene altogether that's just a simple dominant and recessive gene. So we're not going to worry about the RH factor right now. That's what that's called, RH factor, and that's whether you have that RH or not. All right, so the gene is I, and I has three forms: IA, IB, and little I. And you'll notice that we have two dominants here, okay? And what we're going to have is a codominant situation plus this recessive, all right? So when we're writing genotypes for blood type, if you are type A, you can be homozygous dominant and have IA, or you can be heterozygous and be IA, little I, right? We'll get to the donation and receiving in just a second, okay? Let's get the genotypes done first. All right. If you are blood type B, same scenario. You can be IB, or you can be IB, little I. So you can be homozygous dominant or heterozygous. If you are blood type AB, you can be IA, IB. And that's the codominant blood type. That's where you're expressing both the A and the B gene. If you are type O, then you have two recessive alleles. Okay, so you're little I, little I. So those are the genotypes for blood type. So you got all these possibilities for genotypes that you didn't have before. It's not three different genotypes. Now you've got 1, 2, 3, 4, 5, 6 different genotypes because you've got three alleles. What does this mean? What this means is if you are blood type A, you have type A receptors on your cells. So your blood cells have type A proteins on them. If you're type B, you have type B proteins on them. If you're type AB, you have both A and B proteins, and if you are type O, you don't have any of these proteins. Now, the reason why this is important is, remember, these proteins are markers that tell the immune system that the cells belong to you. And so these proteins are important for blood donation because the proteins have to match your immune system. So if you are somebody who is type A, we'll start here at type A, you can donate to somebody who is type A. You cannot donate to somebody who's type B because what would happen is your A proteins on your cells would cause them to have an immune system response to your blood, and that could kill someone. If you wanted to donate to somebody who's AB, you could also donate to somebody who is AB because they have A proteins on their blood, and so their immune system recognizes the A protein and it's no problem. If you cannot donate, to somebody who is type O, though, because again, type O would be somebody who doesn't have any proteins on their blood, and that protein would upset their immune system. All right, you can receive from type A, and you can receive from type O. The reason why you can't receive from type AB is because the B proteins on the AB blood would upset your immune system. All right, so for type B, you can donate, just like with type A, you can donate to type B or AB for the same reasons, and you can receive from B or O. If you are type AB, you can only donate to somebody who is AB. Okay, and again, the reason is if you try to donate AB blood to somebody with type A, then the B proteins are going to upset their immune system. If you tried to donate AB blood to somebody who's type B, the A proteins are going to upset their immune system. So you can really only donate to one person here. But you can receive from type A, type B, type AB, and type O. Okay, so you can receive from any other blood type, which means you are the universal acceptor if you have type AB blood, meaning you can accept blood from anybody. If you are type O, you can donate, notice, to A, AB, B, or O, but you can only receive from somebody who's type O, because if you receive blood that has any protein markers on it at all, then you're going to have an upset immune system. All right, so you are the universal donor. All right. Now, this is important for blood donation purposes so that you don't upset somebody's immune system. All right, now let's talk about the implications of this. First of all, the blood banking idea was instituted by Dr. Charles Drew, and this was because people used to notice several things. First of all, you could donate blood to somebody who was in distress, and sometimes that person would be okay, and sometimes it wouldn't be okay. And again, this was because of blood type incompatibility. Okay, so prior to Dr. Drew's idea of collecting blood, typing blood, and then storing it to give to the right people, you could just give somebody blood from somebody else, and half the time it would work, and half the time you'd kill somebody. So this is an important advancement. Okay, so this is for donation and transfusion purposes. There is another issue that arises with blood donation that you do need to be aware of, and that's with moms and babies. Okay, when a mom is pregnant with a baby, her blood and baby's blood don't mix. They come really close together in the placenta, but mom and baby's blood don't mix. All right, the problem is when mom gives birth, the placenta separates. Okay, and when the placenta separates, and the placenta is that disc-shaped organ that lays on the side of the uterus and supplies the baby with all the stuff it needs, all the oxygen and food and nutrients from mom, and it also gets rid of all the waste from the baby. So at birth, the placenta bleeds. Whoops. And that's the first time baby's blood is loose in mom's system. And in a situation where you've got a mom, say, who is type A, but a baby that she's carrying is type B, mom can have an immune system reaction to that baby blood now that's loose in her system. So what doctors will do is test the baby's blood and test mom's blood right away. They test mom when she goes to the hospital, and then they test baby right after the baby's born. If there's a blood type incompatibility, and by that I mean what you saw on that first slide, if you've got a situation where mom's getting contact with blood that she really shouldn't, then they give mom a drug to suppress her immune system so she doesn't have an immune system reaction. Now, the immune system reaction is probably very small because we're talking about a very small amount of placental blood getting into her system. The problem doesn't arise then. It arises with the next pregnancy. If she has another pregnancy with the same daddy, what can happen is she'll miscarry the next time. So if mom carries another baby that has an incompatible blood type, she'll have a miscarriage, and she won't ever be able to get pregnant again. So that's why it's really important that we know about blood type as well, because mamas want to have more than one kid sometimes. All right, let's look at a couple examples of how this plays out. You've probably heard this used in terms of paternity testing, and this was the old school paternity test. The thing is, there are cases, blood type can never be used to establish paternity. In other words, it's never going to be able to tell you that somebody is definitely the father. And here's an example of why. Okay, so you've got a woman with type A. So we know she's type IA. Okay, for the moment, let's just skip on the other part of her blood type. Okay, so she's got IA. We know that her child is type O. So her child is little I, little I. All right. Now, if baby is little I, one of these little i's had to come from mama. So now we know mama's heterozygous. Okay. And we're asking, can a man with type little I be the baby's father? Okay. Well, you can probably see it, but you can also put this onto a Punnett square. Don't forget, you can do Punnett squares with these problems. IA, little I. Okay, so here's our Punnett square. And you can see that two of the babies on this Punnett square, these two right here, could potentially be this baby, okay? But does that mean he's the father? The answer is no, and here's why. He's not the only guy in the world with this genotype, right? Anybody who's blood type O could technically be the father. Also, anybody who's heterozygous for blood type A or B could be the father, right? Anybody with IA little I could father a child with this woman that has this little I from, right? Anybody who is type B and heterozygous could be the father, right? Because daddies can pass that little I along instead of the dominant trait. So blood type can never say, yes, with 100% accuracy, this man is the father. It can suggest that he could be the father, and that's all we can do. If you want to establish paternity, you've got to do a DNA test in this case. All right, so when blood type can be used to exclude a man as the father, then it can do so completely, and here's an example like that. So we've got a woman with type A, so we have her, here she is, IA, and she has a child who is type O, so same scenario as before. So she's IA little I, okay? Now, she's claiming that a man who is type AB is the father, okay? Now, some of you are looking at that and can see it right away, okay? Obviously, this came from the mama, and some of you are saying, okay, well, there's no way that this other little I could come from this man, and you're right. But you can also put this onto a Punnett square. So IA, IB, here's the daddy, little I, okay? Whoops, sorry, the mama is IA, little I. There we go. All right, so when we put this onto a Punnett square, when you fill it out, you'll notice you'll get a child that's type A, a child that's type AB, another child who's type A but heterozygous, and a child who is B and heterozygous, but no little I. So this guy is not the father of the baby. This is a rare case where you can actually use this to exclude somebody as being the father. Most of the time, it's the situation like we saw in the last example. Okay, so most of the time you're not going to get a situation like this, but occasionally, and usually one of the parents is AB, and usually when that happens, there's a way to disprove that somebody is the father. But again, a DNA test would confirm that with 100% accuracy.